Hello everyone, in this video I'll be explaining on the topic human immune response and hypersensitivity reactions. Coming first to the mechanism of humoral immune response, first is neutralization of bacterial toxins and neutralization of pathogens by neutralizing antibodies. Here, most of the microorganisms use molecules present in their envelope or cell wall and gain entry into the host cell. The antibodies bind to the microbial envelope or cell wall molecules and colonizing the host. Second mechanism is opsonization of the pathogens. Antibodies coat microbes and promote their ingestion by the phagocytic cells. The process of coating particles for phagocytosis and their engulfment is called as opsonization. The molecules that coat microorganisms and enhance phagocytosis are called opsonins. Next mechanism is activation of complement by classical pathway. The complement activation in classical pathway requires antigen-antibody complex and hence eliciting humoral immune response. The next mechanism is antibody-mediated cellular cytotoxicity. Antibody will bind to the surface of virus-infected cell and is recognized by IgG receptors present on the surface of cytotoxic cells such as macrophages and natural killer cells, and the infected cells is killed. Antibody-dependent cellular cytotoxicity process can also kill helminths. In this case, IgE is the antibody involved and eosinophils are the killer cells. Coming to the scope of cell-mediated immune response, it also participates in various immunological functions such as delayed hypersensitivity reaction, immunity in various infectious diseases, which includes bacterial infections like tuberculosis and leprosy, fungal infections like histoplasmosis and blastomycosis, protozoal infections like leishmaniasis, trypanosomiasis, viral infections like the measles and mumps, and also it participates in the transplantation immunity and graft versus host reaction, in pathogenesis of certain autoimmune diseases like the thyroiditis and encephalomyelitis, and also against immunity of cancer. Coming to the mechanism of cell-mediated immune response, first is afferent stage. In this stage, the antigen is bound to the macrophage surface membrane and then it is ingested and processed within the macrophage, the epitope of the MHC complex membrane, and then presented to T cell. Second mechanism is central stage. The stage begins with activation of T cell, that is, helper T cells, cytotoxic T cells. In order for the central stage to occur, it requires the activation of T cell, that is, helper T cell and cytotoxic T cell. The first signal in the activation process is the interaction of antigen and the MHC protein with T cell receptors specific for that antigen. When the T cell receptor interacts with antigen MHC protein complex, the CD4 protein on the surface of helper T cell also interact with class II MHC protein. In addition to the binding of CD4 proteins with MHC class II protein, other proteins also interact to stabilize the contact between the T cell and antigen presenting cell. A second co-stimulatory signal is also required, that is B7 protein present on the antigen present in cell must interact with CD28 protein present on the helper T cell. If co-stimulatory occurs, interleukin 2 is produced by the helper T cell, and this step is crucial to producing helper T cell capable of performing its function. After T cell has been activated, a different protein called CTLA4 appeared on the T cell surface and binds with B7, replacing CD28. The interaction of B7 with CTLA4 inhibits T cell activation by blocking interleukin 2 synthesis. Next step in the mechanism is efferent stage. Helper T cell release biological mediators which activate macrophages, enabling them to kill intracellular parasites, and these lymphokines activate cytotoxic T cells and natural killer cells, destroying the target cells. Coming to the classification of hypersensitivity, first is type 1 hypersensitivity, which is also called as anaphylactic reaction. It is IgE mediated reactions. Type 1 reactions occur in two forms, that is anaphylaxis and atopy. In anaphylaxis, it occurs when sensitized individuals come in contact with a shocking dose of antigen. IgE molecules bind to the surface receptor of mast cells by their Fc end, and then when a shocking dose of the same or immunologically related antigen is given, the antigen combines with cell-bound IgE antibody on mast cells. The antigen-antibody complex stimulates mast cells to release mediators that cause clinical manifestation of anaphylaxis. Next is autopsy, which runs in families. This includes these individuals have tendency to produce reagent antibody in unusually large amount. Reactions occur at the site of entry of the antigen, like inhalation of pollen affects the lungs, resulting in bronchial asthma, ingestion of milk, food, fish, egg, etc., causing gastrointestinal disorders, and contact lenses lead to local allergy. The antigen combines with a cell-bound IgE antibodies fixed on the surface of mast cell, and this antigen-antibody complex stimulates these cells to release mediators, resulting in clinical features of atopy, example, food allergy, dust allergy, and drug allergy. Type 2 hypersensitivity is also called as cytotoxic reaction. This reaction is mediated by IgG or rarely IgM antibodies directed against antigen on the surface of cells, resulting in cell damage. Example, hemolytic disease of the newborn. Type 3 hypersensitivity reaction, it is characterized by deposition of antigen-antibody complex in tissues leading to tissue damage. Two typical type 3 hypersensitivity reactions include Arthur's reaction due to relative antibody excess and serum sickness due to relative antigen excess. Arthur's reaction in this, Arthur's observed that with repeated subcutaneous injection of normal horse serum into rabbit, the initial injection were without any local effect, but with later injections, intense local edema and hemorrhagic necrosis developed. This type of reaction is called as Arthur's reaction. Next is serum sickness. This serum sickness appears following a single injection of high concentration of foreign serum. Type 4 hypersensitivity reaction, it mediates that by T lymphocyte which on contact with specific antigen release lymphokines that cause biological effect. Type 4 hypersensitivity occurs within 48 to 72 hours, and two types of delayed hypersensitivity reactions are recognized. First is the tuberculin type, and next is the contact dermatitis type. Next is measuring immune functions. In this, evaluation of humoral immunity can be done by B cell count by flow cytometry and immunoelectrophoresis. And in case of test for evaluation of cell-mediated immune response, it is divided into in vitro test and in vivo test. Under in vitro test, first is the skin test for presence of delayed type hypersensitivity. Normal person responds with delayed type reaction to skin antigen of Candida, streptokinase, streptodornase, etc. because of past exposure to this antigen. Absence of reaction to this skin test suggests impairment of cell-mediated immune response. Second is skin test for the ability to develop delayed type hypersensitivity. Normal person readily develop the reactivity to simple chemicals. Example, dinitrochlorobenzene applied to the skin. When the same chemical is applied to the same area 7 to 14 days later, they respond with delayed type reaction, whereas immunocompromised person fail to develop such delayed type hypersensitivity reactions. Under in vivo test versus the lymphocyte blast transformation, when sensitized T lymphocytes are exposed to specific antigen, they transform into large blast cells, which increase DNA synthesis. The blast transformation is of two types, specific and non-specific. Non-specific blast transformation occurs when exposed to certain mitogen, whereas specific blast transformation occurs in the presence of plant extracts like phytohemagglutinin. Next is macrophage migration inhibitory factor. This occurs when culture T cells are exposed to the antigen to which they are sensitized, and the effect can be measured by observing the reduced migration of macrophage in the presence of the factor compared with the level in control. Next is enumeration of B cell, T cell, and its subpopulation. It can be done using a machine called fluorescent activated cell sorter. In these, the cells are labeled with monoclonal antibody tagged with fluorescent dye, such as fluorescein or rhodamine. Single cells are passed through the laser light beam, and the number of cells that fluorescence are identified.